Perfect. Okay, good. Okay. So what I'd like to tell you about is some of our recent work having to do with how microbiota talk to T lymphocytes, and try to put this in the context of the little bit that's known about how microbiota affect the anti-tumor T cell mediated immunity, and tell you about one of these projects in our laboratory. These are my disclosures over here. Just moving on, we know that there's a great amount of influence of the intestinal microbiota on anti-tumor immunity. And these three papers just a few years ago really crystallized the importance of the microbiome, showing that for cancer immunotherapy, such as CTLA-4 or PD-1 blockade, to function well, an animal must have a microbiota. Germ-free mice do not have effective anti-tumor responses, and antibiotics really blunt the anti-tumor responses as well. So there are many papers that have appeared since then, and I'm not going to dwell on those. And some of these have described various metabolites made by different components of the microbiota. All I can say at this point is that there isn't yet a consensus mechanistically as to what is going on. And I'll show you the system towards the end of my talk, the system that we've been trying to work out in order to see if we can get some general, if we can come down to some general principles of how the microbiota function here. But what is also has gotten a lot of attention is that fecal microbiome transplantation can be partially effective. And this is a couple of studies published now almost 4 years ago that showed that there are some people who had failed immunotherapy, and then following fecal transplantation with material from individuals who had responded very well, some of these do respond quite well, as shown over here, but others do not respond. And it may have to do with the quality of the microbiome that they are receiving, but we still do not have a good idea of how this may function. So how might this be working? Well, what we know is that in the intestine, along the length of the intestine, where you have an enormous diversity of microbiota, typically, that increases as we go from the proximal to the distal intestine, you can have different types of microbes. We work largely with bacteria, but of course, there are also viruses and fungi and protozoa that can also contribute to the to the entire immune environment in response to the microbiome. But we can pretty much divide the bacteria into three different classes. They're the ones that we think of as being really just simply mutualistic and non-pathogenic and potentially beneficial. And the model system that we use for this is segmented filamentous bacteria, which induces a homeostatic type of T cell that makes interleukin-17 and interleukin-22, reinforcing the intestinal barrier. So these are regulatory type Th17 cells. At the other end of the spectrum, you have potentially pathogenic bacteria that can make toxins and can be invasive, such as Citrobacter rodentium, which is the model in the mouse. This typically induces a Th1, Th17 type of response. And then in between, you can have what are known as pathobions, which is what we work with, which are bacteria that are typically mutualistic, but under homeostatic conditions, but under certain conditions. The ones we use are, for example, when interleukin-10 is limiting, these bacteria can become pathogenic. And these bacteria can induce both a pathogenic type of Th17 cells, as well as regulatory T cells and follicular helper cells, as I will show you. So in addition to the microbiome, of course, the gut is where the nutrients are absorbed, and the immune system must also face the enormous antigenicity that is provided by antigens in the diet. And this typically occurs in the more proximal intestine. And I'll show you some of our recent work on this as well, because there needs to be a regulatory type response against dietary antigen to prevent inflammation from occurring in that setting. So just to then summarize, what I'll tell you about is our work on the pathobiont Helicobacter hepaticus, and also some of our recent work with food antigen. Both of these induce regulatory T cells. In the case of H. hepaticus, the Tregs keep in check potentially inflammatory Th17 cells that can also be induced by the Helicobacter hepaticus. And when the Tregs fail, either for intrinsic reasons in the Treg function or for the inability to induce Tregs due to the microenvironment, that can lead now to the expansion of inflammatory Th17 cells and potentially to an inflammatory bowel disease like Crohn's disease. And in the case of food, the antigens also induce Tregs, and these typically keep in check a variety of other potentially inflammatory T cells. The most common that we think about are Th2 cells, which are involved in various allergies, including food allergy, but also Th1 and Th17 cells can be involved, such as in celiac disease. That's also a breakdown of tolerance in the intestine. So that can lead now to a variety of different kinds of allergic diseases, not only locally in the gut, but also systemically. So our goal is to try to understand what are the cellular targets of both microbiota and food antigen, and eventually identify molecular targets, and trying to understand how this affects the induction of different kinds of T cell programs. So the place where this kind of induction occurs, of course, is in the lymph nodes that drain the various tissues. In the case of food, the upper intestine; in the case of microbiota, throughout the intestine, but largely in the large intestine. And draining lymphatics bring in antigen-presenting cells that pick up the various antigens, typically through a chemokine gradient that involves the chemokine receptor CCR7. And these antigen-presenting cells, largely dendritic cells, then localize to the paracortical region where naive T cells are present. And those naive T cells can now be primed and receive signals to differentiate to go in different directions. And as I mentioned, these can be pathogenic type of Th17 cells, and they express ROR gamma T, as well as homeostatic Th17 cells that can make interleukin-10 and have regulatory-like functions and also express ROR gamma T. But they're also induced or peripheral Tregs that express FOXP3 and often express ROR gamma T as well in the intestine. And I'll show you some of this. And there can also be induction of follicular helper cells, which are BCL6 positive cells that express also CXCR5. So how is it that these different programs can be elicited by different microbes and different bacteria and different food antigens? So there's a model that's been promoted for a number of years as to the maturity or activation state of the antigen-presenting cell or dendritic cell, such that different kinds of antigens or bacteria in this case, excuse me, may program this antigen-presenting cell in a certain way so that it makes the products to direct the T cells after they are primed to adopt the various types of programs. At the other end of the spectrum, there's a possibility that there are dedicated antigen-presenting cells, and that each of these is engaged by a antigenic entity to now program a naive T cell to adopt one of these different fates that I mentioned. And in between, there's also the possibility that there's some plasticity between these antigen-presenting cells, or that they can have that they have the possibility, the ability to induce a particular subset of T cells, depending on the kinds of signals that they receive. So this was a hypothesis that we started out with, and when Ranit Kedmi who was a very talented postdoctoral fellow now at the Weizmann Institute, when she took this on. And she adopted a system that had been developed by Mo Shu, another fabulous postdoctoral fellow who is now at NIBS in Beijing. And the system that Mo had developed was this Helicobacter hepaticus system that I had mentioned to you, in which mice are colonized with Helicobacter. These are SPF mice that can be wild type or mutant mice, so we can study the microenvironment effect here. After a week, we introduce into these mice T-cell receptor transgenic T cells that had been developed by Mo that are specific for a peptide of Helicobacter associated with MHC class II. And we can label these cells with CFSE or some other cell tracker, so we can track them in vivo as they proliferate. We can look at day 3 in the C1 mesenteric lymph node that drains the large intestine. And then we can look later, a week or two later, in the lamina propria as well to track these cells. So you can see if we now just gate on the donor-derived T cells, which are isotype marked, you can see that after 3 days, they have proliferated. They have upregulated FOXP3. They've also upregulated ROR gamma T. And these are these peripherally induced regulatory T cells now specific for Helicobacter hepaticus. And if we now look about 10 days later in the large intestine, we see these double positive cells, regulatory T cells here that typically make up about 2/3 of the cells. And then another third of the cells here are BCL6 positive cells that are follicular helper cells. So this is typically what's seen. Now, if we make mutations in which the Tregs cannot develop, such as a mutation in the transcription factor CMAF, then we don't see this, but we see just Th17 cells expanding. But what Ranit decided to do was to look in mice that are defective for the chemokine receptor CCR7 in what we thought would be dendritic cells, using CD11C Cre to knock out a conditional allele of CCR7. So you can see in a heterozygous mouse, if we now look in the large intestine, you see largely Tregs here in different mice and very few Th17 cells that are ROR gamma T positive. On the other hand, in mice lacking CCR7 in these cells, you see the loss of the Tregs and the expansion of the Th17 cells. And after a few weeks, you see accumulation in the intestine, the large intestine, of mononuclear infiltrates, and eventually these mice will develop a colitis. So what surprised us here was that there was still, even with the loss of CCR7 in dendritic cells, there was still the differentiation of pathogenic Th17 cells. And these were even observed when we had a full germline knockout of CCR7, but even more surprising, when we used CD11C Cre to knock out MHC class II, we also saw the loss of the Tregs and the proliferation of the Th17 cells. So what that told us then is that for these pathogenic type of Th17 cells, which I should say express typically not only ROR gamma T, but also T-BET, the transcription factor typically seen in Th1 cells, and they can also make interferon gamma. What happens here is that these cells are programmed by a CD11C negative antigen-presenting cell that's also independent of CCR7. And we think this is a monocyte lineage cell, and I'll come back just briefly to tell you where we think this is coming from, and we're trying to identify that precisely. But the focus today will be on what induces these peripherally induced regulatory T cells. And initially, we thought they were type 2 dendritic cells, conventional dendritic cells, that make CCR7. And excuse me 1 second here while I turn off my, my email sounds. I don't know if that was disturbing, but anyway, I'll try to avoid that. So this was our initial thinking, that these were conventional dendritic cells that are CCR7 dependent, and that if we now knock out with either of 2 dendritic cell drivers with Cre drivers, CD11C Cre or ZBTB46 Cre, knock out CCR7, that leads to the loss of the regulatory T cells. and the expansion of the Th17 cells. And we also saw this if we use these drivers to knock out MHC class II, H2M, which is required to displace the invariant chain peptide and replace it with other antigenic peptides presented by MHC class II, or the integrin alpha V that pairs with beta 8 to activate TGF-beta from its latent form into an active TGF-beta. And we know that TGF-beta is essential for the induction of these peripheral regulatory T cells. So all of this seemed consistent with these being a dendritic cell. But then we and two other groups found that when we used a different driver, ROR gamma T-cre, which is typically not expressed in antigen-presenting cells, what we found was the very same kind of phenotype. And this was very surprising. And so we thought initially we couldn't tell whether there were multiple antigen-presenting cells involved, but in the end, it really appeared that there had to be an antigen-presenting cell, and I'll show you why, that expresses ROR gamma T as well as CD11C and ZBTB46. And this would be a novel antigen-presenting cell involved in the generation of these peripheral regulatory T cells. So the three different groups that reported this 3 years ago were, in addition to us, a group of Greg Sonnenberg and also of Sasha Rudensky and Chrissy Brown. Greg's group concluded that there were type 3 innate lymphoid cells, which are known to express MHC class II, that were doing this job. Chrissy's group proposed that there's a novel cell type that they call a theta cell that expresses in some cells the air transcription factor that's also found in thymic epithelial cells and is involved in induction of tolerance in the thymus. And we could not reach a conclusion. Initially, we thought they were type 3 innate lymphoid cells, but then through a collaboration with Jay Gardner at UCSF, we thought that they may also be these air-expressing cells that Jay Gardner called Janus cells that might be involved in doing this. But we didn't really have a tool at that point to really pinpoint which antigen-presenting cell was inducing these regulatory T cells. So what I'll tell you about now is where we think that these cells exist and what they are. So the experiment that Ramit did that told us that there is a distinct cell type that's doing this, a one cell type, was a gain of function type of experiment in which he used bone marrow chimeric mice in which he irradiated the recipient animals and then introduced into them bone marrow from either wild type mice or mice in which MHC class II had been knocked out with CD11C CRE, or mice in which there was a gain of function in which the MHC class II was expressed only in CRE-expressing cells. So we could use CD11C CRE or ROR gamma T CRE. These are mice that were made by Terry Lauford that she kindly shared with us. So we could reconstitute with these kinds of bone marrows, and then after 8 weeks, colonize the animals with Helicobacter, and then use the approach that I described earlier to look at the TCR transgenic cells as well as endogenous T cells to see what might happen. So when we put in wild-type bone marrow, as you might expect, you see the FOXP3 ROR gamma T double positive Tregs and the follicular helper cells down here, which you can see here express BCL6. And when we use CD11C CRE to knock out MHC class II, the Tregs are lost, and instead we see these ROR gamma T positive pathogenic type Th17 cells. When CD11C CRE was used to turn on MHC class II, a stop-lox allele that now can express only when CRE is expressed, you see that we reconstitute the Tregs and the follicular helper cells. And what was really the most satisfying result was that when we use just ROR gamma T CRE, we only get the Tregs and we don't see the BCL6 positive follicular helper cells. So what this experiment conclusively showed was that there are multiple antigen-presenting cells that each of which has a dedicated function. So in the case of the pathogenic Th17 cells, it's a CD11C negative cell type that induces the Th17 cells when it expresses MHC class II. And like I said, we still don't know precisely what this cell is, but we think it's monocyte derived. In the case of follicular helper cells, we know it's a CD11C positive cell that can do this, but CD11C positive cells also can induce regulatory T cells. But ROR gamma T positive cells also can induce only the Tregs here. So we conclude that it's a cell type that expresses both ROR gamma T and CD11C that induces a regulatory T cells. So our focus now has been on trying to precisely identify what this cell type might be. And I told you it's been proposed to be this novel TC, Thetis type cell, or Janus cells, or type 3 innate lymphoid cells. So this is the work that Li Wei, Fu, and Rabbi Upatier took on and benefited greatly from a mouse model set up by Maria Pokrovsky when she was a graduate student in the lab many years ago, but a mouse system that we had not really exploited fully yet. And I'll tell you about that now. So the first thing we wanted to know was whether the transcription factor ROR gamma T is required for these cells to develop and to be able to induce regulatory T cells. I showed you that they're ROR gamma T positive cells, but the function of ROR gamma T was not tested. So what we did was to cross CD11C Cre with a fluxed allele of ROR gamma T and repeat this experiment that I described. And just as I showed before, there's essentially a complete loss of regulatory T cells, indicating that ROR gamma T is required for these antigen-presenting cells to develop or to assume their function. Instead, what we see is expansion of ROR gamma T positive, T-bet positive, pathogenic type of Th17 cells. We see these also if we just look not only at the TCR transgenic cells, but also at the host T cells. And you can see in the host, there's a loss of the Tregs and the expansion of these Th17 cells. So we next wanted to look at the innate lymphoid cell compartment. And I'm sure many of you on this call know about the relationship of innate lymphoid cells and T lymphocytes that have different programs. And this is something that has evolved only in the last decade or so. It's become clear that there are innate type cells that don't have adaptive capacity of rearranging receptor genes that have very similar kinds of programs to T cells, particularly to CD4 T cells. So, for example, the ILC1 express T-bet just like Th1 cells and can also make interferon gamma. The ILC2 are GATA3 dependent, positive like Th2 cells. And then there's a whole slew of different type 3 innate lymphoid cells, including a lymphoid tissue inducer cell that's involved in the generation of lymph nodes and Peyer's patches, as well as tertiary lymphoid tissues. And all of these express ROR gamma T and are dependent on ROR gamma T for their early development and resemble the Th17 lineage or Th17 programs of T cells, including the non-pathogenic and the pathogenic. And the more pathogenic corresponds to these T-bet expressing ROR gamma T positive cells as well. So this takes us back now a decade to when Maria Pokrovsky was in the lab, and she wanted to look at ROR gamma T expression in these different types of cells, as well as Th17 and gamma delta 17 cells, and as well as thymocytes, where ROR gamma T we first found to be important. And she adopted the method that had just been developed of ATAC-seq. This is bulk ATAC-seq, in which she was able to look at in vitro differentiated Th17 cells, as well as ex vivo cells, either from the intestine, Th17 cells. We used a reporter here to isolate these cells, gamma delta cells, type 3 innate lymphoid cells, as well as thymocytes. And you see in each of these cell types, there are unique chromatin accessible regions. Like in the thymocytes, you have this peak over here, whereas there are also these regions here, +6 and +7 kb in the ROR gamma locus that are distinct for Th17 cells versus type 3 innate lymphoid cells. So Maria concentrated on this and made knockouts of the +6 and +7 regions. And when she knocks out the +6 region here, you can see there's a loss of Th17 cells, but it doesn't so much affect the 7kb mutation doesn't affect the Th17 cells. On the other hand, when she makes a 7kb mutation, she deletes this region here. There appears to be a loss of ILC3, but we're looking at Rho gamma T expression. So Rho gamma T is expressed much less here. Also in the innate-like T cells, the gamma delta 17 cells, there's also reduced Rho gamma T expression, but there's no effect on Th17 cells. So we decided to look at these mice because they seem that they might be useful for asking the question whether there might be a defect in the induction of regulatory T cells. And indeed, when we now look at the 7kb mutant mice using the Helicobacter system, we basically see this repeated phenotype, the loss of Treg, expansion of Th17 cells that express Tbet and ROR gamma T. And this just shows the really dramatic phenotype that's present here in the Treg loss and the expansion of Th17 and Th1-like cells. So this gave us then two different platforms in which we could perform single-cell RNA sequencing to compare what happens compare these mutants to wild-type mice, looking at lymph node antigen-presenting cells, CD11b, CD11c positive cells, and/or CD11c positive cells in the draining lymph node. And these are the different clusters from the single-cell RNA sequencing that Robbie and Li Wei performed. And we see ROR gamma T in only three populations. The type 3 innate lymphoid cells, of course, a population of cells that expresses neuregulin-1, NRG1, that looks a lot like the fetus cells described by Brown and Rudensky, as well as a population here that are the only ones that express another transcription factor called PRDM16 that's been described previously to be important in adipose tissue browning. And if we now compare the proportion of these cells in mutants and wild type using both the 7kb mutation and the deletion of Rho gamma T using C11C3, we see that the only one that's affected here in the mutant is this PRDM16 population that's reduced now, but there's no real effect on ILC3. And if we now look at the full knockout in the CD11C cells, there's a complete loss of this entire cluster here. We don't see it anywhere, and we think it's just not developing, and no effect on type 3 innate lymphoid cells you see here, or in this on this TC-like population shown over here. So that really pointed us towards PRDM16, this transcription factor that might be important in this particular function. And we're very fortunate that there are some good antibodies for intranuclear staining of PRDM16. So Li Wei developed a panel of staining looking at Rho gamma T expression and MHC class 2 over here. So these are the cells we're interested in that are antigen-presenting Rho gamma T positive cells in the lymph node. The Rho gamma T negative cells here, you can see, are largely these conventional dendritic cells that express CD11C. So we could look at these different populations and break them down now into those that express PRDM16 and are negative for CXCR6 that Chrissy Brown had shown is expressed on type 3 innate lymphoid cells. So we can look at the ILC3 here and these what we think are dendritic cells that are tolerance-inducing dendritic cells. And these are their properties. These PRDM16 cells that are positive here also express the highest level of Rho gamma T. They do not express IL-7 receptor, which is, of course, a marker of lymphoid lineage cells. But these IL-7 receptors expressed on the ILC3 in green here. But they do express CD40, which is, of course, found on dendritic cells and is involved in the importantly involved in the interaction of activated T cells with the APCs. And they do not express CXCR6 as opposed to type 3 innate lymphoid cells. And at some time in their development, they have to express CD11C. Only part of them express CD11C here, but ILC3 do not express CD11C or CD11B. And moreover, we then also did single-cell ATAC-seq on the different populations from the lymph node. And these are just some markers for dendritic cells. for dendritic cell lineages, particularly the conventional dendritic cells, IRF4, which is very important for CDC2s, CLEC9A, which is expressed on CDC1s, typically, and you can see it's most open on CDC1s, and FLT3, which is involved in dendritic cell development. And what you can see is that the PRDM16 positive cells are very similar in their chromatin pattern. This goes throughout the genome to the migratory conventional dendritic cells and not to the type 3 innate lymphoid cells. And that's why we are calling these we're calling these tolerogenic dendritic cells, or we haven't come up with the exact name of this yet. We might call them regulatory dendritic cells, and maybe Miriam can suggest what might be the best name here, because there are a lot of names for different kinds of dendritic cells. But I think functionally, these are dendritic cells that induce regulatory T cells. So what we could do now also is look at PRDM16 expression when we knock out Rho gamma T using C11C Cre. And if we look at the mesenteric lymph node, you can see now that we lose these PRDM16 population in these mutant mice that cannot induce Tregs. And we can also look in the small intestine and also large intestine. And these cells are present in the lamina propria, and they are lost at least for PRDM16 expression. But we think these cells are lost based on the single-cell RNA sequencing in the absence of ROR gamma T. And there's really no effect on the type 3 innate lymphoid cells in these cases. The other thing we're able to do was to use ROR gamma T Cre now to knock out PRDM16. We got a floxed allele of PRDM16 from Bruce Spiegelman, and could knock out these cells. We could knock out this gene in the Rho gamma T APCs. And again, using the Helicobacter-specific system, we basically phenocopy this and see the loss of Tregs and the appearance of pathogenic type of Th17 cells, as shown here. So now I think we have a very clear definition of this cell type, and I can call it maybe a novel type of dendritic cell that is CD11C and ZBTB46 positive, at least at some point in their development, also are dependent on ROR gamma T and PRDM16, much as CDC1s are dependent on BATF3 and IRF8, and CDC2 on IRF4. The precise ontogeny is not clear yet. We think they likely come from a common dendritic cell progenitor. And we're now doing cell lineage tracing and lineage stamping to try to precisely identify the progenitor cell. Very briefly, we think a monocyte dendritic cell progenitor likely gives rise not only to these, but also to antigen-presenting cells that can induce the pathogenic Th17 cells. And from both gain and loss of function experiment, we have evidence that CLEC9A is the CLEC9A Cre transgene made by Cayetano Reyes Souza is expressed in these progenitors here, even though they're CD11C negative. We're trying to precisely identify what this antigen-presenting cell might be, but I don't have time to really go into that. So the last part of the story here pertains to allergic responses. And what we noted was in the 7KB enhancer mutant mouse, what we saw was that there was not only the defect in Th17 cells, but we also saw an expansion of GATA3 positive cells, as shown over here in the intestine. So this alerted us to the possibility that there may be also a defect in allergic responses. And of course, both food and microbiota must be dealt with in the intestine. And if there's a defect in the tolerance to food antigen, that can lead to expansion of these IL-4, IL-13 expressing cells and production of IgE and in various kinds of allergies, including food allergy. that in parallel to what can happen in response to the microbiota here. So we decided to look at this a little bit, and using a very well-known model of oral tolerance, in which it is known that if you gavage animals with an antigen before sensitization and then challenge, that can prevent allergic responses. So we first wanted to look using the chicken ovalbumin model at oval-specific MHC class II specific T cells, OT2 cells, transgenic T cells, in this particular model. First looking, see after gavage with ova, is there induction of regulatory T cells? And if you can see in the control here, if we gate on these OT2 cells, that you see that many of these cells are FOXP3 positive with and without ROR gamma T. And there's very little in the way of GATA3 expression here. But in the PRDM16 mutant mice, you can see there's a loss of the Tregs and the appearance of both ROR gamma T positive and GATA3 positive cells here. So we then wanted to look in a model of airway sensitization in which we can prevent airway disease by tolerizing with ovalbumin. And the model has intraperitoneal ova with alum as an adjuvant that is administered. And then a couple of weeks later, one can introduce ovalbumin intranasally several times, and that can now lead to an allergic lung response unless there is tolerization here. So we could look both in the 7kb mutant mouse as well as the PRDM16 mutant animals. And here with the 7kb wild type and mutant mice, you can see that there's much more of an inflammatory infiltrate in the mutant mouse in this model in which tolerization was attempted. And if the animals are not tolerized, there's an accumulation of eosinophils in the lung that are Siglec-F positive and CD11c low. And these cells are prevented from accumulating if there is oral tolerization prior to that. But in the mutant mice, you can see the oral tolerization fails completely. And that now leads to the accumulation of eosinophils as well as T helper 2 cells. This is now in the PRDM16 mutant, where we basically see the same phenotype, and also accumulation of Th2 cells and production of IgE that cannot be controlled. And we then also used a second model, which is more of a food allergy type model in the Black 6 mice. The Black 6 mice are not ideal for looking at type 2 types of responses, but we can, it's better to do this in BALB mice, which we are now trying to generate as well. But in this particular mutant mouse, now the 7kb enhancer mutation, what we can show is that there's a loss of tolerance to intragastric sensitization. So in this case, there's tolerization or not, then there's ovalbumin is introduced with cholera toxin as a sensitizer intragastrically. So this is now a oral kind of challenge of sensitization. And then the challenge is systemic with intraperitoneal ovalbumin. And in mice that are not tolerized, there is a very rapid loss of body temperature that is observed within the first half hour in these mice, and oral tolerization prevents this. But in the mutant mice, you see that they actually do more poorly from the very outset in not being able to be tolerized, and there's really absolutely no effect with or without tolerization in these animals, and you see the loss of tolerization for production of IgE specific for ova, and also IgG1 specific for ova. And in the last couple of minutes here, I'm just going to summarize this and then just mention briefly our tumor model. But what we find now, if we look both at public data sets as well as data from a mesenteric lymph node, from lymph nodes from a healthy patient a healthy donor that we obtained, is that these PRDM16 positive cells can be observed both in human lamina propria colon and ileum, as well as in donors of tonsil donors, and these are PRDM16 Rho gamma T positive cells. And when we compare their gene expression pattern to those in the mouse, we find that there are a number of genes that overlap that you can see over here between what we call the tolerogenic mouse and human DCs that are not shared with conventional dendritic cells. And some of these genes are listed up here that are highly expressed both in mouse and human. And we're now trying to generate reagents to be able to study the human tolerogenic antigen-presenting cells. So just to summarize this then, what we now can say is that there are these different dedicated APCs that can be accessed by some bacteria, like Helicobacter hepaticus, and each of these APCs is dedicated to a particular program of T cells. And similarly with homeostatic type of T cell inducing bacteria like SFB, these actually use a more conventional type of dendritic cell to induce homeostatic Th17 cells. And in the case of food, what we've shown is that either the same type of APC or a very similar type of APC that's dependent on Rho gamma T and PRDM16 induces peripheral Tregs to food antigen. But Th2 cells certainly can also be induced by the food antigen, as well as Th17 cells. So typically, these peripheral Tregs keep these other responses in check, so you don't get the pathogenic type of responses. But if there's a defect in the induction of these peripheral Tregs, now these pathogenic cells can arise, causing either inflammatory bowel diseases or various kinds of allergic responses. So this raises many different questions. I list a few of them here. It's you know, are there, we can say is that there stereotype T cell responses induced by different kinds of bacteria. We don't really know how they access distinct type of antigen presenting cells. Is it the niches that they occupy? Is it particular recognition molecules that they use? It's really something that needs to be looked at. So I should talk to you about the tolerogenic APCs. We don't know if there are differences in these APCs in different regions of the gut. We also, one of the really interesting possibilities that they may also tolerize to self-antigen, and that's certainly what Chrissy Brown and Sasha Rudensky suggested with their study on their fetus cells. But I should say that these cells are present throughout lifetime. They are very abundant proportionally early in life, but then they are retained even as they are their proportion goes down later in life. So they can do their job both early in life and late in life. These cells are very rare in the intestine and mesenteric lymph node, and at least in what we can extract from these tissues. Now that we'll be able to image them, we'll hopefully be able to tell where they are, but we don't really have an explanation for how they can be so efficient. They resemble the conventional dendritic cells, but not ILC3 in chromatin accessibility and gene expression, and we like to know what their precise ontogeny is. So the really exciting implications for us is that genetic or environmental perturbations may lead to imbalance in these kinds of APCs. Are there variants, gene variants involved in their developmental and functional programs that could be involved in autoimmune and allergic diseases, as well as resistance to pathogens? And finally, can these be accessed therapeutically in some way, either generated from iPS cells or targeted in vivo to tolerize? So these are all questions that we'd like to answer in the future. So the last 5 minutes here, I'm going to just turn to cancer and to how the gut microbiota may influence immune responses to cancer, which is really the topic of this entire lecture series. But I wanted to give you background on how one studies T-cell induction by the microbiota. And we also know that the nervous system can also impact on this, and there's a lot of recent excitement about innervation of cancer, gut-brain interactions in controlling various kinds of immune responses. So I won't have time to go into this. Hopefully, some of the other speakers will delve in this at some point. But I'll tell you quickly about the model system that we use, which is a system that was developed in collaboration with Talis Papagiannakopoulos, our colleague at NYU, which is initially we studied an orthotopic lung cancer model that has a mutation in P53 and the mutant RAS. And we also knocked out one of the DNA repair genes, MSH2. So these tumors can accumulate antigenicity, accumulate mutations that can be antigenic, immunogenic. And these tumors express a luciferase, so they are implanted in the lung, and we can track them. We can track their growth through the luminescence. What Robbie Upatier, who was set up the system to study it, did was to show that with anti-PD-1, there was reasonably efficient control of these. But if you treated the animals with antibiotics and just ampicillin was sufficient, that basically eliminated the efficacy of this PD-1 blockade. We also, at the same time, were studying the role of microbiota in autoimmune disease. And Varsha Raghavan, when she was in the lab, was using EAE, experimental autoimmune encephalomyelitis, as the model. And she, like others previously, showed that when antibiotics were used in animals that had been immunized with a myelin protein, MOG, was that the paralysis could be prevented with either vancomycin or ampicillin. There was really effective prevention here. So what she decided to do was to isolate a large number of ampicillin-resistant bacteria from humans, and then test to see whether they could restore this sensitivity to MOG immunization. And she tested many different types of bacteria in which she colonized mice that had been treated with antibiotics. And what she found was that a couple of strains of Bacteroides vulgaris were very efficient in restoring the disease, but other bacteria, like the related Bacteroides ovatus, had no effect whatsoever. So what Robbie decided to do was to see whether he could use the same system to look at his tumors. And what he found was that these different strains of Bacteroides vulgaris, again, in the presence of antibiotic, of ampicillin, were very efficient at preventing the tumor growth. And the surprise here was that this was even in the absence of anti-PD-1 therapy. On the other hand, the Bacteroides ovatus had really no effect whatsoever, very mirroring what we observed with the microbiota, and this shows here the luminescence. And what he showed is that the protection is dependent on T cells, both CD4 and CD8 T cells, and it could be transmitted by blood exchange, and I'll show you the model quickly that we use to look at blood exchange. It could also be replicated in mono-associated mice, so it's not just an effect of something else growing in the presence here of the ampicillin. And it was observed with multiple tumor types in the C57 Black 6 mouse, which was quite, in a way, quite surprising. And it implied that it's not due to some kind of cross-reactivity for self-antigen with the microbiota, if it had an effect on so many different tumor types. So I'll show you just one experiment in which we tested this. There's also a possibility of antigen non-specific activation of host innate responses that result in activation of host or tumor reactive T cells, and we favor this possibility here. as well as the possibility of cryptic antigens being induced in the tumor, rendering the tumor more immunogenic. And that might explain why multiple Black6 tumors are doing this. And we know that endogenous retroviruses can be reactivated in tumors. There's a lot of work like Sebastian Amigorena's work showing that these can produce tumor-specific antigens that can be recognized by the host immune system. Just one experiment I'll show you to look at the possibility of molecular mimicry here. We know that we can use molecular mimicry in a model in which, for example, if we express SFB bacterial antigens in a tumor and then colonize with SFB, you can get a mucosal Th17 response to the SFB, but now those cells can actually migrate into the tumor and elicit in there a Th1 response that controls the growth of the tumor. So it is possible to have molecular mimicry there, but we don't think that B. vulgaris functions through mimicry. In an experiment that we did, first of all, we know that CCR7 is required, of course, for being able to prime a T-cell response, and we and others have done this kind of experiment before. In a CCR7 knockout mouse, you no longer can see the protection with the B. vulgaris. So that's not surprising because the dendritic cells cannot drain to the lymph nodes, and there's also, of course, a defect in the T cells the naive T cells going into the lymph node. So the experiment that we did was in collaboration with Scott Manalis's group, because Scott's group set up a way of doing a parabiosis with mice in different cages. So you don't have to saw to really tie the mice together. You can have them separated by basically having catheterization of the jugular and carotids in these mice, and you can connect them between different cages with a peristaltic pump and even collect blood from these animals. And we typically would do 60-minute sessions every day for 8 days. And if we do that, what we can do is we can implant the tumor in this mouse, and we can have a second mouse in which we put the microbiota, or we can put the B. vulgaris in this animal here. And we showed that after 8 days, there's very good control of tumor growth if we have B. vulgaris in these animals, but only if we have B. vulgaris. Otherwise, we don't see the control of the tumor. And in this case, what we wanted to do was to ask whether CCR7 might have a role here. So what we did was to colonize the mice that were either CCR7 negative. So now there couldn't be good priming of the T cells in these mice, but they have the B. vulgaris. And then this mouse, the healthy mouse, has a tumor here. I mean, the wild-type mouse has a tumor implanted. Both of the animals are on ampicillin. And when we do this, what we see is that there's now no effect on the ability to control the tumor growth. Granted, we did this with a small number of mice. These are very difficult experiments to actually set up. But what this implies is that it isn't T cells that are primed by the microbiota, by the B. vulgaris, that now travel to the tumor to do their job, because the T cells cannot be effectively primed in those draining lymph nodes. So it really, we think that you do not have molecular mimicry here, but some other mechanism is involved here. So this leads to a number of different questions here. Are the T cells from B. vulgaris required for anti-tumor effect? We think probably not. Are there differences in plasma metabolites? We do find differences in metabolites, particularly tryptophan metabolites, when we colonize with B. vulgaris versus B. ovaris and are testing those. We'd like to know what the differences are in the composition of the tumor microenvironment and lymph nodes from these tumor-bearing mice. We have some early suggestion that there may be differences in expression in the tumor of otherwise silent elements, including endogenous retroviruses, but we'll see if that turns out to be the case. So the last thing we want to know whether B. vulgaris could restrict or reverse tumor growth in a therapeutic model of lung cancer. And here we used, in collaboration with Thalys and Ray Pillai, this model they developed, in which it's an autochthonous model with a mouse that has it's a Cas9 expressing mouse with the KRAS and P53 inducible mutation, can be induced with CRE. So we use a virus now that expresses CRE, as well as provides guide RNAs for MSH2 to knock out the repair process pathway here. So when we do this, put this into mice at 12 weeks, we can look at baseline tumor growth in the lung using MRI, and then we can intervene here with colonization, ampicillin treatment and colonization, and follow the development of the tumor here. And so what we do here, you can see the baseline scan, and you can see that 7 weeks post-colonization, now you can see the growth of the tumors here. So this is at 12 weeks, I believe, and then you can see the continued growth at 7 weeks with these multifocal tumors in the lung of these mice. And if we now look at 5 weeks after colonization and 7 weeks after colonization with either B. vulgaris or B. ovatus, you can see that there is still growth of tumor even with B. ovatus, but it is considerably retarded compared to the control with ampicillin alone or B. ovatus. So this is done without any checkpoint immunotherapy. We're repeating these experiments now with added immunotherapy, hoping that we can actually stop the tumor growth, not only retarded, but we are encouraged that this is a model that will allow us to actually look at a therapeutic setting. So we think that B. vulgaris colonization can work to some degree therapeutically to retard the growth of the tumor. So I've gone almost an hour now, so probably too long. I hope there's still time for questions. I mentioned those who have really been the key people involved in this work. We collaborate with Scott Benalis's group with these parabiotic mice, and these are the folks who helped us with the tolerogenic dendritic cell story. So I'd like to thank you very much for your attention, and sorry if I've gone a little bit too long here. So.